All right, we can get started for today. First, I want to welcome everyone for joining us for today's webinar. My name is Yuan Tian. I am a content marketing scientist at Biocytogen Boston Corporation, and I'll be moderating today's webinar. And I'm joined by our speaker today, Dr. Dong Han. He's a senior scientist at Biocytogen, where he also leads the neurological disease team. So before joining Biocytogen, Dr. Han earned his PhD in medical sciences from Kanazawa University in Japan, where he focused on biochemistry and type 2 diabetes. After that, he moved to Boston and worked as a postdoc at Joslin Diabetes Center at Harvard Medical School. He then continued his research at UMass Boston, where he served as a postdoc and later as a research assistant professor. Now at Biocytogen, Dr. Han leads the development of the neurological and behavioral platform using both chemical-induced and genetically modified animal models. So for today's webinar, Dr. Han will introduce the basic concepts of neurological disorders, their impacts on the brain and behavior, and how animal models are used to uncover disease mechanisms and evaluate therapies. Before we dive into today's presentation, I would like to remind our viewers that the Q&A box is open. So feel free to drop your questions there, and we will address as many questions as we can at the end of the presentation. With that, I'll hand it over to Dr. Han.
Thank you very much, Yuan, for the kind introduction. Hello, everyone. My name is Dong Han. I'm a senior scientist at Biocytogen. It's my great honor to have you join our webinar. So today we have a group of fantastic panelists who are well-trained in the field of neurology research here to support this webinar and answer any of your questions or concerns. So without further delay, I'm going to present our recent advances in animal models of neurological disorders. So neurological disorder is a quite a broad term. According to World Health Organization, neurological disorders are diseases of the central and peripheral nerve systems. So these disorders include epilepsy, Alzheimer's disease, and other dementia, cerebrovascular disease, including stroke, migraine, and other headaches disorders, multiple sclerosis, Parkinson's disease, neuroinfections, brain tumors, traumatic disorders of the nerve system due to the head injury, and the neurological disorders as the result of a malnutrition. So it is estimated that there are globally 47.5 million people with dementia, with 7.7 million new cases every year. So Alzheimer's disease is the most common cases of dementia and may contribute to 60 to 70% of the cases. So a comprehensive study published in the Journal of Lancet Neurology found that in 2021, 3.4 billion individuals, that is the 43% of the world population, is affected by the neurological conditions. The number of disability-adjusted life years, DALYs, caused by the neurological conditions increased to 443 million years of healthy life lost, so making neurological conditions the leading cause of disease burden worldwide. The report listed the top 10 neurological conditions contributing to the loss of health. Therefore, more therapeutic strategies need to be done and developed to help those affected individuals and their families. In this webinar, we're going to focus on the use of animal models in neurological research, specifically the use of genetically engineered mouse models in the research area of blood-brain barrier, Alzheimer's disease, autism spectrum disorders, amyotrophic lateral sclerosis, and Parkinson's disease. So central nerve systems, CNS, is a highly controlled microenvironment to promote its normal functions. The blood-brain barrier, BBB, is formed by the microvascular endothelial cells, lining the cerebral capillaries, penetrating the brain and the spinal cord of the CNS. The existence of the biological barrier at the blood to brain interface effectively separate the brain from the rest of the body. The BBB is playing a critical role in protecting the brain parenchyma from blood-borne agents and providing a significant obstacle to the delivery of the drugs and other compounds into the CNS. Transport routes of the drug molecules across the BBB could happen via the pathways including paracellular and transcellular diffusion, transporter-mediated transcytosis, receptor-mediated transcytosis, and adsorptive-mediated transcytosis. Since the receptor-mediated transcytosis can transport large molecules like insulin and transferrin, it has attracted significant interest from the researchers focused on the drug delivery across BBB. Transport vehicle TV drug delivery technology, which utilizes the receptor-mediated transcytosis RMT pathways on brain endothelial cells, are utilized as an entry point into the brain. The most well-studied RMT target is transferrin receptor TFR, which interacts with transferrin and import iron to the brain. Antibody transport vehicle ATV engineered to bind TFR can increase the brain delivery 20 to 30 times more than the non-RMT binding counterparts. These TFR binding platforms have been used to deliver various large molecule therapeutics to the brain parenchyma, including enzymes, proteins, and antibodies. Another BBB transport target discovered by the proteomic profiling is a CD98 heavy chain. CD98HC hydrodimerize with the various large amino acid transporters that includes the LAT1 and is essential for trafficking these transporters to the cell surface. Similarly, this ATV platform was engineered Fc domain for binding CD98HC that enables the brain delivery of biotherapeutics. So with all the advanced transport vehicle platforms, how can we test their efficacy in vivo? At Biocytogen, we generated a humanized TFR1 knock-in mouse model named BHTFR1 mouse. The exons 4 to 19 of mouse TFR1 gene that encodes the extracellular domain of the TFR1 protein were replaced by the human TFR1 exons 4 to 19, and the intracellular region, which encoded by the mouse TFR1 gene exons 1 to 3, were kept unchanged. So this model can be utilized for testing the binding of a human therapeutics with humanized TFR1 protein and the BBB penetration with an intact mouse intercellular signaling component. mRNA was extracted from the spleen of a C57BL6 mice and the BHTFR1 mice. Mouse TFR1 mRNA was only detected in the spleen of wild-type C57BL6 mice with the primer designed on the exons 1 to 3, and the human TFR1 mRNA was detected only in the homozygous BHTFR1 mice with the primers designed on the exons 4 to 19. So on protein level, bone marrows was collected from C57BL6 and the BHTFR1 mice, and the species-specific antibodies were used for flow cytometry analysis on the TFR1 expression. The results shows mouse TFR1 was detected in the wild-type C57BL6 mouse bone marrow using the mouse-specific antibody. Human TFR1 was exclusively detected in homozygous BH-TFR1 mice, but not in the wild-type C57BL6 mice. Immunofluorescent staining analyses were also carried out in the BH-TFR1 mice. Brain paraffin sections were prepared from an 8-week-old female wild-type C57BL6 mice and homozygous BH-TFR1 mice. The paraffin sections were stained with the DAPI in blue to show the nuclei of the brain cells, anti-mouse CD31 antibody in red to stain mouse microvascular endothelial cells, and anti-human TFR1 antibody in green to show human TFR1 expression. So the results indicated that human TFR1 expression was only detected in the BH-TFR1 mouse and was co-localized with the microvascular endothelial cells. In a co-development project with our client, we intravenously administered anti-human TFR1 bispecific antibodies or control IgG into the BH-TFR1 mice via tailwind. The brain and the serum were collected for in vivo PK studies. So figure 1 shows a higher concentration of human TFR1 bispecific antibody compared with the control IgG in BH-TFR1 mouse brain. Figure B shows a faster serum clearance of a human TFR1 bispecific antibody compared with the control IgG in BH-TFR1 mice. Figure C shows a higher brain-to-serum ratio of TFR1 bispecific antibody compared with the control IgG in BH-TFR1 mice. These results demonstrated that BH-TFR1 is a powerful preclinical model for in vivo evaluation of effective delivery of protein therapeutics. In a very recent elegant study published in the Journal of Science, carried out by the Dr. Deverman's group at the Broad Institute of MIT and Harvard, that using BH-TFR1 mice, they demonstrated their engineered adeno-associated virus AAV capsid BIHTFR1 delivers genes and transduces neurons and astrocytes throughout the whole brain and spinal cord with a much higher efficiency compared to the AAV9. This state-of-the-art study showcased one of the possible applications of a BH-TFR1 mouse model. Another receptor-mediated transcytosis target attracted many attentions is CD98HC. Biocytogen generated a humanized CD98HC knock-in mouse model named BH-CD98HC mouse. The exons 2 to 10 of mouse CD98HC gene that encode the extracellular domain were replaced by human CD98HC exons 4 to 12. RNA was extracted from the kidney of C57BL6 mice and the BH-CD98HC mice. Mouse CD98HC mRNA was only detected in the kidney of wild-type C57BL6 mice, and a human CD98HC mRNA was detected only in the homozygous BH-CD98HC mice. On protein levels, splenocytes were isolated from C57BL6 and the BH-CD98HC mice, and the species-specific antibodies were used for flow cytometry CD98 detection. The result shows mouse CD98 was detected in the wild-type C57BL6 mouse in T cells and B cells. Human CD98HC was exclusively detected in the homozygous BH-CD98HC T cells and B cells. Immunofluorescent staining analyses were also carried out in the BHCD98HC mice. The brain paraffin sections were stained with the DAPI in blue to show the nuclei of the brain cells, anti-mouse CD31 antibody in red to stain mouse microvascular endothelial cells, and anti-human CD98HC antibodies in green to show human CD98HC expression. The results indicated that human CD98HC expression was only detected in the BHCD98HC mice and was co-localized with the CD31 expressing microvascular endothelial cells. So we have a wide range of off-the-shelf, constant breeding animal models for BBB research purpose. The targets are ranging from TFR1, CD98HC, to IGF-1R and LDLR. More valuable to your research, we also have double humanized animal models. If you're interested at any model, feel free to let us know here in the chat box, or if you have a unique model in mind, would like us to generate for you, we're more than happy to fulfill your specific requirements. So now let's switch gear to Alzheimer's disease models. There are 55 million people worldwide suffering dementia, with nearly 10 million new cases increased every year. So Alzheimer's disease is the most common form of dementia and may contribute to 60 to 70% of the cases. Dementia is the 7th leading cause of death globally. So Alzheimer's disease, AD, is characterized by the chronic and acquired memory impairment and cognitive deficits in domains such as languages, spatial temporal orientation, and executive capacity, as well as behavioral alterations, all of which lead to progressive loss of personal autonomy. Clinical signs and symptoms shows amyloid deposition from B PET scan, generalized cortical atrophy shown by the MRI images, and the tau deposition in the tau PET scan images. Alzheimer's disease is marked by extracellular senile plaques composed of amyloid beta, A-beta, the accumulation of intracellular neurofibrillary tangles, NFTs, made from hyperphosphorylated tau protein, neuronal loss, and significant brain atrophy, all contributing to cognitive decline and other AD symptoms. The 3 important genes associated with the AD that includes APP, PSEN1, and ApoE. To assist research in AD, Biocytogen generated the transgenic 5FAD mice, expresses human APP and PSEN1 transgenes with 5 AD-linked mutations, named BTG5FAD mice. The IHC staining shows the amyloid beta expression in the cortex in 3 months and 5 months old of B transgenic 5FAD mice. Another pathogenic factor in Alzheimer's disease is a tau protein. So we generated a humanized tau mouse model named BH-tau mice. RNA was extracted from the brain of C57BL6 and the BH-tau mouse models. Mouse tau mRNA was detected in 57BL6 mice, and a human tau was detected only in the humanized tau mouse models. A preliminary experiment was carried out to confirm the human tau expression in the mouse model of BH-tau mice. So the BH-tau mice were grouped into 2 arms. One was treated with the PBS. The other was treated with the human tau siRNA by ICV injection. After 7 days of treatment, whole brain were collected and subject for mRNA and protein analysis. The results demonstrated that treatment of a human tau siRNA significantly decreased both mRNA and protein level of a human tau in the mouse brain compared to the PBS-treated mice. The results confirmed that the expression of a humanized tau protein expression in the brain of the model. Additionally, we generated a mouse model BH-tau P301S with the full coding sequences of a human tau gene with a P301S mutation driven by a mouse PRNP promoter are inserted into the mouse HEAP11H11 locus, a safe location in the BH-tau P301S mice. So RNA was extracted from the brain of C57BL6 and the BH-tau P301S mice. Mouse tau mRNA can be detected from both animals, and the human tau can only be detected in the BH-tau P301S mouse. And further Sanger sequencing analysis on the PCR product of a human tau PCR, we confirmed that tau mutation from the proline to serine at the position of amino acid 301. Western blot assay on the protein extracted from the cortex and the hippocampus of C57BL6 and BH-tau P301S animals shows that overexpression of a total tau accompanied with the increased phosphorylated tau. Next, we're featuring another important humanized model in AD research, BH-TREM2 mice. The full mouse TREM2 gene coding sequence was replaced by human coding sequence and under the mouse TREM2 promoter. RNA was extracted from the brain of C57BL6 mice and the BH-TREM2 mice, and the mouse TREM2 mRNA can only be detected in the C57BL6 mice. Human TREM2 mRNA exclusively detected in BH-TREM2 mouse brain. Microglia were extracted from both C57BL6 mice and BH-TREM2 mice. Due to the antibody availability at the time, a human and mouse cross-reactive anti-TREM2 antibody was used, and compared with the isotype control antibody, TREM2 expression was detected in both C57BL6 and the BH-TREM2 microglia. Furthermore, in the Western blot assay on the brain lysate from a C57BL6 and the BH-TREM2 mice, human TREM2 was exclusively detected in the heterozygous BH-TREM2 mice that confirms the knock-in was successful. So here we have a current list of animal models for Alzheimer's disease research. They're ranging from classic 5FAD models, APP, and PSEN1 double humanized models, humanized transgenic tau models, humanized TREM2 models, humanized ApoE models, and double humanized AD models with the BBB targets that would make them a perfect tools to test the therapeutics. So please feel free to let us know even you just want to know more about the models or you have your model in mind would like us to develop for you. So next, I will introduce autism spectrum disorder, ASD. ASD is a prevalent and complex neurodevelopmental disorder, which has a strong genetic basis and environmental risk factors exposure causing abnormal neural development. ASD typically manifests in early infancy and childhood. It is characterized by the difficulties in social communications and interactions, as well as repetitive behaviors and restricted range of interests. The mechanisms of ASD are complicated and are linked to the transcription factors and the translational mechanisms. The key pathways like RAS, ERK, and the PI3 kinase mTOR, mutations in the transcription-related proteins such as MECP2 in Rett syndrome and the UBE3A in the Angelman and the duplicate 15q syndrome, and in translation-regulating proteins, for example, P10, ADNP, EN2, TSC1, TSC2 in tuberous sclerosis, and FMRP in Fragile X syndrome. These are associated with ASD development. To study Rett syndrome, we generated a BMECP2 knockout mice that the exons 3 to 4 of mouse MECP2 gene were deleted. Proteins were isolated from C57BL6 and the BMECP2 knockout mice. Western blotting results showed that MECP2 protein expression were successfully knocked out in the BMECP2 KO mice cortex and hippocampus. Rett syndrome is an X-linked neurodevelopmental disorder, predominantly affecting girls. It is caused by mutation in the gene encoding methyl CpG binding protein 2, MECP2, on the X chromosome. Homozygous female and the males of BMECP2 knockout mice exhibit hypoactive and have a shortened lifespan, dying by 3 to 4 and 10 to 12 weeks of age, respectively. Heterozygous females of BMECP2 knockout mice are viable and fertile and utilized in many studies. An important target in studying Fragile X syndrome is FMR1 gene. So we generated a FMR1 knockout mouse model by deleting exons 3 to 17 of mouse FMR1 gene. The protein expression analysis shows that FMR1 protein was successfully knocked out in the cortex, hippocampus, testis, spleen, and lung of the BFMR1 knockout mice. Fragile X syndrome, FXS, is a monogenic disease caused by mutations in the FMR1 gene. Although FXS occurs in both male and female patients, most animal studies adopt male FMR1 knockout mice. Male BFMR1 knockout mice show significant hyperactivity in the open field test. In details, there are significant higher total distance traveled, average speed, and the number of crossing in the BFMR1 knockout mice. So the behavioral study shows that BFMR1 knockout mice demonstrated Fragile X syndrome phenotype hyperactivity. Now I will introduce amyotrophic lateral sclerosis, ALS, also known as Lou Gehrig disease. It is a fatal neurodegenerative disease that affects the nerve cells that control muscles. Symptoms include muscle weakness, cramps, and twitching in the limbs, hands, feet, and shoulders and tongue. Speech problem, drooling, involuntary emotional expressions, and fatigue. ALS pathogenesis involves several pathways, including proteostasis failure, disrupted RNA metabolism, mitochondrial dysfunction, neuroinflammation, and excitotoxicity. ISOD1, the first gene linked to ALS, remains a key focus in treatment research. Despite over 40 randomized trials, there's no treatment have been proven effective and significantly slowing down the disease progression or improving the survival. Riluzole, which has a modest effect on the survival, is currently the only disease-modifying drug approved by all the major drug authorities. Here is our model list for ALS studies. Please let us know if you would like us to know more about your research or you have a novel targets to target this devastating disease. Lastly, I'm going to introduce Parkinson's disease. PD is a chronic brain disorder that causes movement problems, mental health issues, and other health concerns. Symptoms that include stiffness, slow movement, difficulty with the balance and coordination, and the loss of a smell. Other symptoms include mental and behavioral changes, sleep problems, depression, memory difficulties, and fatigue. Environmental and genetic factors contribute to PD by impacting the key biological pathways, such as the mitochondrial dysfunction, oxidative stress, alpha-synuclein aggregation, and the clearance of the abnormal proteins. Currently, there's no cure for the PD. Treatment focuses on the symptom management with the dopaminergic drugs, which elevates the motor symptoms but don't halt the disease progression. We're currently working on launching our initial models to assist the PD research. Last but not least, we have mouse models in assisting rare neurological disease, such as spinal muscular atrophy and Huntington's disease. We're hoping these models can be assistant to you and accelerate your research. So we are a well-established CRO company, provides a comprehensive disease models. Besides the genetic engineered animal models we just introduced, we'll also provide chemical-induced neurological models, such as multiple sclerosis and acute epilepsy models, and surgical models such as ischemic stroke. And we're constantly developing more animal models and increase our capabilities. To assess the behavioral phenotypes of the animal models, we established the scratching assay, open field, rod, pain test, treadmill, grip strength, balance beam, Y-maze, and novel object recognition. For histology capability, we established the histopathology, cytopathology, and molecular pathology. We also can facilitate antibody screening, efficacy evaluation, non-GLP safety evaluation, and translational medicine evaluation. So we are constantly developing new animal models to facilitate and speed up the research needs for the neurological disorders in the field of ALS and Huntington disease. And there's a constant growing list for our model build. So here is a complete product list for our neurological disorders models. So if there's any models that you'd like to inquire more, that please feel free to reach out to us or to any of our business development manager, and then we're happy to share the data and more information with you. So please allow me to introduce our company briefly. So our company was founded in Worcester, Massachusetts in 2008. And then first of all, we established the gene editing platform in Beijing. And also we developed the humanized immune checkpoint mice, BNDG mice. And after the models were developed, we introduced our pharmacology service platforms that can serve our customers from the utilization of animal models to their PK/PD needs. And upon those platforms, we also established the antibody discovery platform and RenMap platform launched in 2019. And also we launched our RenMap high heat platforms. So we're a fully integrated, comprehensive service provider to support your needs for the target to IND. So we have a disease target selection from the gene edit by the gene editing service. We provide more than 4,500 customized models, and then we provide preclinical tumor and disease models with 1,700 off-the-shelf models. And we also support the preclinical validation studies to develop and deliver more than 2,500 projects to our customers. So with that, I will express my sincere appreciation to your participation to this webinar. Thank you.
So the first question, how do you have any phenotypic data from the AD models?
From the Alzheimer's disease models. Yeah, thank you so much for your question. So as you'll see that the disease models for Alzheimer's, we freshly developed those models, and they're under the expansion of a colony. And as you know, some disease required age to have the phenotype on site. So we're currently working on those phenotype data with our behavioral platforms. And then once the animals have reached their phenotype age, and then I think we'll share the data with you. Thank you so much.
Second question that came in, the genes associated with ASD, autism spectrum disorder, are the same as cancer-related genes. So does ASD also make humans susceptible to any cancer types?
I can take this one. So, ASD and cancer do share some genetic overlaps, such as RAS, ERK, PI3AGT, and the Wnt pathways. While these genetic overlaps do exist, the relationship between both ASD and cancer susceptibility is not necessarily straightforward, right? Some research shows that there is some overlap and there is some susceptibility, and other research shows the exact opposite. So I guess the best answer to this is, while there is some genetic overlap between the two, it's not universally understood or known, or it means that folks with ASD are at a higher risk for cancer. However, there is some research that shows that certain genetic mutations, like the P10 and NF1 gene, for example, do make patients with ASD a little bit more susceptible to specific cancers.
For ALS model, how is your SOD1G93A model differ from SOD1 model that is typically used? Do you have any biomarker and behavior data?
Currently, those models is still under development. So that's a very good question. So I will consult with our model development team, and then I'll get back to you on that question. Thank you so much.
Next question, considering neurological diseases take a long time to develop in humans, can we expect humanized animal models will close the gap to the discrepancy observed in clinical trials in neurological animal models and human disease?
That is a very good question and a very important question. So currently, we're generating these animal models, and then we're going to test the phenotypes of the animal models while they're at early age. And more important is to let them age, and then perform the phenotype study again to show if there are any differences as the data that are acquired when they were young. So that will clear the discrepancies on the age. Thank you.
Okay, next question. It is great to see the knock-in validation data on the mRNA and protein expression in the BHTFR1 mouse models. Does it show any phenotypic difference comparing to the wild-type animals?
Oh, that's a great question. So many of our customers ask that same question. So they want to utilize that animal models to show the delivery of their therapeutics, but they're concerned of humanized animals can unintentionally to insert some phenotype, present some phenotypes. So internally, we're testing the humanized TFR1 animals on all of our behavioral platforms, compare with their leader mates or wild type mice, and then so preliminary data shows that they don't have any phenotypes on the treadmill running, rotoroids, and balance beam. So we're going to continuously working with the humanized TFR1 animal models on all the behavioral platforms that we have, and then we'll showcase the data together. Thanks so much for the question.
Okay, the next question. Do you have models for certain rare genetic neurological diseases, such as hereditary spastic paraplegia?
So that's a good question. So I don't think currently we have that in mind on the list, but yeah, thank you for raising that point. So we'll contact our developmental team, and then we'll discuss the possibility of develop that model. Thank you so much. It's a great point.
Can I jump in briefly for that as well?
Yes.
If you're interested in seeing that model, we can also support you with developing that model if you would like to work with us in creating a custom animal model. The cornerstones of Biocytogen is our gene editing technology. And so we are able to generate these custom models for you. Often our clients come to us mentioning a specific gene they're interested in. And I know there's a couple different genes associated with the disease mentioned, so we can definitely set up a meeting to discuss this further if you have a certain target you want to talk about.
Okay, thanks, Dawn and Taylor, for answering this question. Next question, what possible roadblock you foresee in starting the phenotype for the rare disease, and how do you plan to identify and develop it?
You mean the rare neurological disease?
Yeah, because I guess those behaviors, those are not like some common behavior tests. You will need a special behavior test to test the behaviors of those rare diseases. Like, how do you plan to identify those phenotypes and develop the behavior tests, I guess?
Yeah, that's a great question. That's a great question.
I can jump in on this one if you like, Dr. Hung.
Thank you, Amrit.
Yeah, no problem. So currently with rare diseases, right, there's a lack of animal models that exist. So the biggest roadblock here is to find suitable animal models in order to do these studies where we're looking at disease mechanisms and that are complex or even human specific, right? So a really good approach in order to get over this roadblock is to create these humanized mice models where we can go out and test different genes to see whether or not they're actually affecting a certain phenotype that comes out. And that's something that Biocytogen actually does really, really well. Is what the models you see aren't just the models we're working on, right? We have a large range of different animal models that we're coming up with and constantly testing to see how we can push forward the research of rare diseases that you're talking about.
Thanks, Amrit. Next question. Do you have fMR1 knockout mouse model behavior data compared to white-type mice?
I can hop in here. So we do have some open field data that we can share with you. If we could connect after the meeting, one of our BDs would be able to support you with sharing that data, and we could set up a meeting to discuss it further. But we have compared the knockout to wild type Black 6 mice in open field.
Yeah, and we're also developing a lot of new behavior platforms. So we have the capability to do a lot of behavior tests. And we're in the progress of doing more. So if you're interested, feel free to contact us. We'll hand over the new data set for you. Just feel free to contact. Next question. Are there any genetic mutations that can lead to AD and PD, I guess Parkinson's disease development in young mice as compared to wait for old age?
I can jump in on this one as well. So for our humanized tau P301S mice, typical phenotypic onset was about 6 months for those. We are currently developing a V2 version of it where we should expect those inclusions to come about 2 to 3 months earlier than what we've seen. So it is a model that we are currently developing right now. So if you're interested in it, feel free to reach out to one of us after, and we can keep you in the loop as these new models are developed and as well as when more data comes out.
All right, one last question in the Q&A box right now. Is there any novel model for AD that have been developed by Biocytogen other than what we already have, like the 5X FAD model? And if so, has it already been validated? This is for Alzheimer's disease.
I think we're constantly developing new models. So our strength. First of all, we're definitely developing the classic models first, and then we're going to. We're going to develop more models, and then more importantly is we develop the double humanized models and triple humanized animal models that serve the specific requirements of the research needs. And from there that we can expand our model list exponentially. So we're constantly looking for looking through the literatures and then looking for new targets to develop models, and also we're doing the double humanized or triple humanized animal models to serve the community.
Okay, thanks, Don, Taylor, and Amrut for answering these questions. I think that's all we have today. Thanks everyone online for participating. Just so you know, this webinar will be available to watch on demand if you did miss anything during today's stream. And also, please don't hesitate to visit our website to learn more about our projects and services. We also have more information to share in the Biocytogen YouTube channel, where you can find more details about our products or services and also our previous webinar recordings. So feel free to check those out as well. And also reach out to our team through our website if you have any questions about anything that was discussed today, or if you have any inquiries about our products. So with that, have a wonderful rest of the day, everyone, and we hope to see you again soon. Thank you.
Thank you so much.